overexpressed in cancer or duplicating cancer. They have a mechanism to stop the P16. P16 do a lot of things, okay? It will be able to stop the angiogenesis, cell spreading, telomerase activity, stop cancer cell growth, stop the VEGF expression. So their impact on CDK is far beyond just a cell cycle itself. It also do something to all these important characteristics. P15, very similar like P16, was regulated by MYC or promoter methylation. They could be negatively regulated by, the positive regulated by TGF-beta or MEZ1, which is the important partner of the MYC to do the job just like P16 does, okay? You don't have to memorize this, but I think it is just try to open the door for you to explore all these important cyclin-dependent kinase activity, inhibitor activity. Again, just like P15 and P16, P21 was positively regulated by many signals, including P53. We talk about that. The P53 is tumor suppressor because it's a genome guardian. We'll talk about that in the third lecture. P53 is a genome guardian. What it does is protect our genome's integrity. The reason for protecting genome integrity is because when the cell receives DNA damage, what we need to do is stop the cell cycle first, so we could repair it, right? Just like many people ride a motorbike on the street. When your motorbike has some trouble, you stop at the roadside first. So you try to figure out how you're going to repair it. So P53 do the job by stop the cell cycle, by inducing P21, okay? Again, this particular CDK inhibitor was regulated by many types of proto-oncogene, including the MYC we talk about, including the HER2. HER2 stands for human EGF receptor type 2, okay? This is a particular proto-oncogene was overexpressed in many types of cancer, including breast cancer. Breast cancer has 4 types, okay? And I hopefully after you finish the lecture here, you get some sense about it. Let me talk about this breast cancer. HER2 stands for human EGF type 2 receptor. It's accounting for 30% of the breast cancer. So breast cancer, you could normally categorize them as an ER-positive type of cancer or HER2. ER stands for estrogen receptor, okay? Some of the breast cancer patients, they have ER-positive, we call ER-positive breast cancer. Some of the cancer, they have the HER2-positive cancer, so we call it HER2-positive. And then another one, so ER usually also have PR, progesterone receptor positive, PR positive. That's one group. The second group is HER2 positive. The third group is called triple negative cancer, meaning that this type of breast cancer do not have ER, PR, or HER2. Okay? I want to give you this brief introduction because the breast cancer, when the pathologist identifies a patient's breast cancer sample, when the pathologist find out if the person is ER positive, then we were using hormonal therapy to stop the ER signaling pathway. If the person is a HER2 positive, we were using the monoclonal antibody against the HER2, such as using Herceptin for stopping the HER2 signaling pathway. The triple negative one is the one disease difficult to deal with. Because we could not using hormonal therapy, we could not using monoclonal antibody against HER2. So this become a challenge for today's therapeutic strategy for triple negative. But I just want to introduce you quickly because we talk about HER2. We'll talk about HER2 later, about HER2's impact on P27. So you get a feeling here HER2 promoter methylation, and another inhibitor to regulate p27 stability called JEB1. And p27 also under a lot of positive regulators. And these positive regulators, usually they are tumor suppressor genes. For example, P10, okay? When you study cancer, you always heard about this particular cancer tumor suppressor gene was particularly mutated called P10. Another one called VHL stands for von Hippel-Lindau. It's an E3 ligase involved in degradation of HIP-1 alpha. HIP-1 is a hypoxia-inducible factor. Okay, so these are all tumor suppressor genes. The green are tumor suppressor genes, and they causing the elevation of p27 to stop the cell cycle. We move on to talk about p57. p57 is very interesting. Okay, p57 will receive signal from prior Q, and then they have an impact in regulating DNA replication, for example, by binding pCNA, by causing the replicated senescence, and involved in muscle differentiation by stabilizing myelin. Okay? p57, unlike p21 and p27, usually these are expressed through both alleles from our father's side or mother's side. You, myself, the p57 gene only expressed from mother's side. So this was called genomic imprinting. I don't know if any one of you know about genomic imprinting, but this is very important genetic regulation of certain genes. Okay? Usually, every one of us have two copies of genes. One from father's side, one from mother's side. Okay? Father's side is called paternal allele, and then maternal allele. Usually, both sides will get expressed. But when there's a genomic imprinting happen, only one side got expressed. p57 is the one only expressed from our mother's side. Many one of you here, p57 is the one only expressed from mother's side. Father's side usually do not express. Okay? So this makes the p57 very vulnerable. If you ever heard about the so-called two-hit hypothesis, the two-hit hypothesis means because we always have two copies, right? Two copies of gene, no more function, we need two copies. Okay? If a cancer cell want to, for example, p53, we want to make p53 totally lost of function. From father's side, from mother's side, we lost the function. Okay? But if only father's side lost function, we still have mother's side still functioning. Okay? So meaning that they still could work, although maybe less efficient. But the cancer, when they're doing the developmental process, we want to make sure father's side or mother's side totally got inactivated. So this is so-called two-hit hypothesis, meaning that paternal allele, maternal allele, we all want to wipe them out. But imagine p57, we only express from mother's side. Make it so easy to just inactivate the one allele from mother's side because the father's side already not expressed. So you just pay attention to some of the genes, they are expressed from different alleles, okay? Some of them only expressed from father's side, okay? When you do genetics, you can pay attention to about this. Okay? So now you get a feeling about all these different cycling CDK, including CDK inhibitors. Again, the CKI genetics or biologists are very interested to understand their biological function. So they try to do the knockout, okay? The knockout mice if the scientists knock out P21, okay, or knock out P27, interesting enough, those phenotype of these knockout mice, they are very normal. Usually, you don't see any defect during the developmental process, which means that P21 or P27's biological function could be replaced or complemented by other CDK inhibitors. But unlike P21 and P27, P57 knockout, remember this is the one who is genomic imprinted, and when we knock it out, the P57, now you have a lot of trouble, okay? These particular knockout mice has a cleft palate, has a gut system defect, has a bone defect, etc. Meaning that P57's biological function cannot be replaced by either P21 or P27. Okay? So just to share with you some of these important gene regulation. And then these are very important aspects because when we want to study cancer, sometimes we pay attention to about these important defects in cancer. I want to sort of look into the biochemistry part about P27. P27 was identified in 1994. Okay? So you could imagine that at that time we didn't know much about CDK inhibitors. And I was very fortunate to be able to participate in this identification of this particular gene. So at that time we're using the so-called affinity column purification. Cyclin CDK was built up for affinity column purification. Basically, we hook up the cyclin E with the so-called histidine column. And then incubate with the nickel beads. The histidine is known to bind to these nickel beads, so they will form an affinity column. And we pass through the cell lysine through these affinity columns. The molecule will be bind to this affinity column. And we only try to elute them out with nickel. Some of the protein got